At 200 meters below the ocean's surface, the last trace of sunlight disappears, not dimmed, not reduced to a pale suggestion, gone, completely, permanently, without exception or seasonal variation. The light that reaches the surface in abundance, that fills shallow water with blue and green, that drives nearly every form of biological energy across most of the ocean, ends here. Below this depth, the water is the same temperature by day as by night, the same color, which is no color at all, only black pressing in from every direction, from above and below, and sideways through the water column, uniform and total and ancient. This boundary has no name that most people have heard. It is not a wall. It is not a line on any map. It is simply the depth at which the last photon of usable sunlight loses its remaining energy to the water and ceases to exist, and below it, the ocean descends in permanent night for thousands of meters further. On the far side of that boundary, something is growing. It has been growing for a long time, longer than the oldest tree anyone has stood beside, longer than the oldest building anyone has entered, longer perhaps than any structure a human hand has made or touched. It grows slowly, a few millimeters in a good year, in water that sits between 4 and 12 degrees Celsius, under pressure that stacks hundreds of meters of seawater above it like stone. It has never required sunlight. It has never had any use for it. The darkness is not something this organism tolerates. The darkness is simply the condition of its existence, as ordinary and unremarkable to it as air is to the organisms that live inside it. The organism is a coral, not the coral that comes to mind when that word is said, not the branching staghorn and table coral of tropical reefs, not the flame-colored fans swaying in warm, clear water, not the sun-drenched formations photographed from above. This is something built on a different logic entirely, assembled over centuries in cold black water by animals that feed on what drifts down from a sunlit world they have no direct connection to. Its name is Lophelia pertusa, though scientists who study it most closely now call it Desmophyllum pertusum. The renaming does not change what it is. It is a colonial coral, meaning that thousands of individual animals, each one a small soft polyp, live together in a shared calcium carbonate structure. They have built themselves, cupping themselves into white stony chambers called corallites that interlock and branch and accumulate over time into something that can span several meters of bare rock face in the darkness of the deep Atlantic. Each polyp is a small cylinder of tissue, open at one end, ringed at that opening by a crown of tentacles. The tentacles are pale. The carbonate cup the polyp sits inside is white. Everything here is white or near to it, not because this is a biological strategy related to light, but because the calcium carbonate that all corals secrete to build their protective chambers has that color by default, and in a world where no light exists to reflect, color is a quality that means nothing at all. The tentacles extend into the water when conditions are right. They are armed with small specialized cells called nematocysts, coiled spring-loaded structures that discharge on contact and paralyze small organisms that brush against them. The polyp has a mouth at its center, and what the tentacles catch, the mouth receives. The polyp does not photosynthesize. It does not contain any photosynthetic partner. Inside the cells of tropical corals, in those warm and shallow reefs that depend entirely on sunlight, there live microscopic algae called zooxanthellae, embedded in the coral tissue, producing sugars through photosynthesis and delivering up to 90% of the coral's energy from sunlight alone. Lophelia pertusa contains none of these. There are no algae in its tissue. There is no photosynthetic machinery of any kind. The coral is purely an animal, feeding as animals feed, catching what comes to it in the current. This is the biological distinction that everything else rests on. Tropical reef-building corals, genera like Acropora, the branching staghorn, and Poritace, the domed boulder coral, are confined to water shallow enough for sunlight to penetrate in useful quantities. That means no deeper than roughly 100 feet, in warm water, within a band centered on the tropics where sea surface temperatures stay above about 18 degrees Celsius for most of the year. Without the zooxanthellae and without sunlight to power them, tropical corals cannot produce the energy they need to secrete calcium carbonate fast enough to build and maintain reef structures. The light is not incidental to the tropical reef, it is the engine. Remove it, and the system collapses. Lophelia pertusa has no engine of that kind to lose. The darkness below 200 meters is not a constraint. It operates around. It is simply where the water is cold enough, the currents consistent enough, and the food supply reliable enough, and those are the only conditions that matter to it. The coral grows in water between 4 and 12 degrees. It grows at depths between 200 meters and more than 4,000 meters. It grows on continental shelf edges, on the flanks of seamounts, at the entrance to fjords, along the ridges where ocean floor topography disrupts the flow of water and concentrates the organic particles drifting down from above. It grows slowly and steadily, and without any relationship to the position of the sun. A single Lophelia polyp, extending its pale tentacle ring from its white calcium carbonate cup into 4 degrees of black water, is doing something that requires no revision to the known laws of biology. It is simply an animal eating, but the accumulated result of thousands of polyps doing this over centuries is a branching calcium carbonate framework that provides structure and shelter to hundreds of other species, that rises meters above the surrounding seafloor, that persists for thousands of years as a physical archive of ocean chemistry and planetary history. The scale of what emerges from this simple process is one of the least known ecological facts about the ocean. On the Norwegian continental margin, off a coastline, more associated in most minds with forest and snow and the light of the midnight sun, Lophelia reefs spread along the seafloor at depths where fishing vessels work and where trawl nets have occasionally torn through the branching framework. Not because anyone knew the reefs were there, but because for a long time, very few people were looking. The reefs on this margin are among the best studied in the world, precisely because Norway was one of the first nations with both the offshore infrastructure and the scientific interest to send instruments and remotely operated vehicles into its own deep waters. What those instruments found, when they found it, did not fit the assumption that reefs were a tropical phenomenon, a product of sunlight and warmth. The Norwegian reefs are extensive, they are dense, they are old, and they are made entirely without light. The cold water off the Norwegian shelf is not an empty place. The Atlantic water that flows northward along the European margin carries nutrients and organic particles that have traveled far, and where the shelf edge creates topographic relief, those particles concentrate. The corals that have established themselves here have been responding to that delivery for a very long time. Individual reef structures on the Norwegian margin have been dated to thousands of years of continuous growth. Some sections of framework are dead, white carbonate skeletons from which the living tissue has long departed. But the skeleton persists, and on the upper surfaces of that dead architecture, new recruits have settled and begun growing. The reef is not one organism. It is a continuous process, generations deep in permanent darkness. Off the western margin of Ireland, along the slope where the relatively shallow waters of the continental shelf give way to the deep Atlantic, there are coral mound provinces of a different character entirely. The Belgica Mound Province, its name taken from a research ship, not a landscape, contains more than 60 individual mounds, some of them rising more than 100 meters above the surrounding seafloor. These are not rock formations, they are not volcanic structures. They are the accumulated skeletal remains of Lophelia pertusa colonies, built up over tens of thousands of years, mixed with sediment trapped within the branching framework, compressed over time into structures of considerable size. The coral polyps that built the base of these mounds are long gone. Their carbonate remains, though, endure. And on the surface of that accumulated past, at this present moment, living polyps extend their tentacles into the cold, dark water of the deep Atlantic and continue the process that produced everything beneath them. Below the Irish margin, below the Norwegian shelf, the same species appears again, off the coast of New Zealand, on the New Zealand shelf, and in the deep water of the surrounding ocean. Cold water coral communities have been documented in the southern hemisphere, on seamounts in the Atlantic, Pacific, and Indian Oceans. Lophelia and related genera have been found anchored to the hard volcanic rock of peaks that rise from the deep ocean floor into slightly shallower but still completely lightless water. In the Mediterranean Sea, at depths where the water is warm by surface standards, but cold and still by the standards of sunlit shallow habitats, cold water coral communities persist on rocky outcrops along the margins of the basin. The distribution of this coral is not a regional curiosity, it is global. What this means, taken together, is something that most accounts of the ocean's biological richness do not adequately convey. The typical picture of a coral reef, the one that appears in documentaries and conservation discussions and the imagery of warm water biodiversity, represents one kind of reef in one kind of environment built by one biological strategy. It is real and it matters, and the animals and plants and microbes that inhabit tropical reefs are genuinely extraordinary. But there is another kind of reef. It exists in every major ocean basin. It reaches depths that no human has visited without a vehicle built to withstand extraordinary pressure. It covers an area of seafloor that recent estimates suggest may equal or exceed the total area of all tropical reefs combined, and the surveys that would more precisely establish this have barely begun. The two systems share a name and share a structural logic. Both build complex three-dimensional frameworks from calcium carbonate. Both create habitat for hundreds of other species. Both accumulate over long time into structures that shape local ecology. But they are built by different biological means in different physical worlds from different starting conditions. One requires a star, the other requires only cold water, a reliable current, and time. There is no hierarchy between them. There is only difference, each one complete. In the Norwegian fjords, something even more unusual occurs. Lophelia pertusa, a species typically associated with depths of several hundred meters or more, has been found growing at depths of 30 to 40 meters in fjords including the Korsfjord, the Trondheimsfjord, and the Sognefjord, 30 meters. In a fjord where the surface is lit by Nordic sky, where in summer the water above glows with the long subarctic afternoon, the cold, dense Atlantic water that flows along the fjord floor at that shallow depth creates a local environment, cold enough, dark enough, and stable enough for Lophelia to grow almost within reach of a surface diver. The darkness there is not the darkness of depth, It is the darkness of cold water flowing beneath warmer surface layers, shaded from above, isolating a strip of seafloor in conditions that belong, physically, to the deep ocean. The coral does not know it is close to the surface. It knows only temperature and current and the presence of food. Those conditions are met, so it grows. There is a particular quality to imagining this, the fjord surface catching winter light, the water column descending 30 meters through cold and increasing darkness, and there at the bottom, in a darkness that is functionally the same as the darkness 400 meters offshore and 300 meters deeper, a Lophelia reef spreading across the sediment. The species is doing the same thing it does in the abyssal dark. It is simply doing it closer to the sky. No part of the coral's biology is oriented toward that sky. The sky is irrelevant to it. What matters is the cold water pressing in from all sides, the small organisms and organic particles carried in the current, the calcium and carbonate dissolved in the water that the polyps extract to build their chambered skeletons. Everything the coral needs is in the dark water immediately around it. Nothing it needs is anywhere above. To understand how different this is from the biology of tropical reefs requires holding one comparison clearly. A tropical coral of the genus Acropora, the fast-growing branching coral that builds much of the framework of reef structures in the Indo-Pacific, grows in water above 18 degrees Celsius, in water no deeper than roughly 100 feet, whereas in water where sunlight penetrates clearly enough for the zooxanthellae in its tissue to photosynthesize. The energy those algae produce, sugars made from sunlight, carbon dioxide, and water, supplies the coral with up to 90% of its caloric needs. The coral's growth rate reflects this. Acropora can extend its branches at rates of 10 to 20 centimeters per year under good conditions, an order of magnitude faster than Lophelia pertusa manages in the cold dark. Remove the sunlight from a tropical reef, cloud it permanently, bury it under sediment, move it to depth, and the zooxanthellae cannot function, the energy supply collapses, and the coral dies. The tropical reef is a solar-powered structure. Its richness and its growth rate and its visual exuberance are all expressions of the energy flowing into it from above, from a star 93 million miles away. Lophelia pertusa is not powered by that star. Not directly, not at all, in the sense that matters biologically. The organic particles that drift down to deep reef depths from the sunlit surface ocean do carry energy that was once captured through photosynthesis far above. But by the time those particles reach the coral at 400 or 800 meters, the connection to sunlight is indirect, dilute, time-delayed. The coral does not respond to light in any way. No behavior changes with the sun's position. No feeding rhythm corresponds to day or night at the surface. The coral lives in a world where those concepts have no purchase. There is no day. There is no night. There is only the cold and the current and the slow arrival of food from above. Below the photic zone, which ends at around 200 meters, the ocean enters a regime called the mesopelagic, and below that the bathypelagic, and below that still deeper zones with their own names and their own particular conditions, decreasing temperature, increasing pressure, permanent absence of light. At 200 meters, the pressure is already 20 atmospheres, 20 times the pressure at the surface. At 400 meters, where many cold water coral reefs are found, the pressure is 40 atmospheres. At 1,000 meters, 100 atmospheres. These are not conditions that require mention because they are hostile to the organisms that live within them. They are simply the conditions. Lophelia pertusa is not struggling against the pressure any more than a person walking at sea level struggles against the weight of the atmosphere above. The organism evolved in these conditions, or in conditions very like them, over a long enough time that the pressure is as fundamental to its physiology as water temperature or salinity. The coral is not surviving the deep ocean. It belongs to it. What cold water coral reefs ask of the person trying to understand them is a kind of quiet revision, not of any one fact, but of an underlying assumption about where life in the ocean concentrates and on what terms. The assumption is that the sunlit surface ocean is the place where biological complexity gathers most densely, and that below the photic zone, the ocean becomes progressively emptier and simpler as depth increases and light decreases. This assumption is wrong, or at least seriously incomplete. The seafloor at 400 meters, where a Lophelia reef spreads across several hundred square meters of rock, is not empty. It is not simple. It supports communities of organisms, fish, crustaceans, echinoderms, worms, sponges, bacteria, of a kind and density that challenge the idea that complexity requires sunlight as its prerequisite. The revision does not require abandoning the importance of sunlight. Most of the ocean's productivity does begin at the surface, in the photic zone, driven by photosynthesis, but the deep ocean is not merely the empty space below that productive surface. It is a world with its own internal logic, its own energy pathways, its own structural complexity built over geological time. Cold water coral reefs are among the most tangible evidence of this. They are not edge cases or exceptions. They are enormous, globally distributed, ecologically rich systems that have been operating continuously for millions of years in water that no sunlight has ever reached. The Norwegian shelf, the Irish margin, the waters around New Zealand, the floor of the Mediterranean, on continental shelf edges, and seamount peaks and fjord floors across every major ocean basin, the same pale branching architecture repeats, the same calcium carbonate chemistry, the same cold water temperature, the same absence of zooxanthellae, the same slow, steady growth and permanent darkness. Each structure is unique in its history and its local ecology, but all of them express the same fundamental biological fact that it is entirely possible to build a reef without light, and that organisms have been doing this in the deep ocean for a very long time. The total area of cold water coral habitat is not yet fully known. The deep ocean is large, and the surveys needed to map it precisely require ships and remotely operated vehicles and time and funding, all of which are finite. What is known is that new cold water coral communities continue to be documented as survey capacity increases, and that earlier estimates of their total extent have consistently been revised upward as more of the seafloor is examined. The current best estimates suggest that cold water coral ecosystems may cover an area of seafloor comparable to or exceeding all tropical coral reefs combined. That would make them by area the dominant coral reef system on the planet, built entirely in darkness at depths most people will never experience, even indirectly, by animals with no relationship to sunlight at all. This does not diminish the tropical reef. The two systems are not in competition. They are not equivalent in the same waters or serving the same ecological functions in the same place. The tropical reef is what it is, a solar-powered structure of extraordinary richness, existing in shallow warm water, built by a biological partnership between animal and alga that has shaped the coastal tropics for hundreds of millions of years. The cold water reef is what it is, a heterotrophic structure of quieter but comparable complexity, existing in cold darkness, built by animals that meet every caloric need through direct feeding at depths where the sun has never reached and never will. Both are real. Both are here at this moment, on this planet, in this ocean, on the warm, shallow reefs of the tropical Pacific. Coral polyps containing their zooxanthellae partners are extending into sunlit water and harvesting the light of a star. At the same moment, in the permanent darkness of the Norwegian margin, of the Irish continental slope, of the seamounts of the deep Atlantic and the Pacific and the Indian Ocean, Lophelia polyps extend their tentacles into cold black water and feed on what the deep current brings them. Neither knows the other exists. They are each complete. The darkness below 200 meters is not a failure of the light. It is not a place where the ocean has given out, or thinned, or become less than what the shallows are. It is a different world entirely, with its own internal economy, its own slow rhythms, its own structures, built over time at a scale most people have never imagined, because the tools to see it are recent and its existence was not expected. The coral building, its white carbonate framework in cold black water at 400 meters depth, small polyp by small polyp, corallite by corallite, colony by colony across kilometers of shelf edge, is not doing something improbable. It is doing something ordinary by the standards of the deep ocean. It is simply living in the dark, under pressure, on cold rock, in still and lightless water that has not changed character since long before any human mind existed to describe it. The coral builds slowly, without sunlight, in the permanent dark below the reach of any star. The reef is there, on every continental margin, in every major ocean basin, in water that no light has entered for as long as those basins have existed. It is vast and old and largely unmapped, and it is there right now, growing in the dark, in the cold, in the deep, at this moment, wider across than most people have ever considered, and older than most structures the surface world has managed to produce. Something that large, existing unseen for that long, in permanent night, asks only to be known about. The knowing does not change it. The cold water reef grows whether or not any mind above the photic zone has ever imagined its existence. It built before anyone looked for it. It will build long after the looking has moved on to other questions. Its whiteness is visible only to the lights of machines descending on cables from distant ships. Without those lights, the white branches are indistinguishable from the dark in every direction, just as they have always been, just as they were when they began, and as they will be when the machines have gone back up and the cables have been coiled and the ship has moved on across the surface. Darkness settling over the branching framework again, water flowing through it, tentacles extended, the reef continuing. The seafloor does not simply end where the continental shelf runs out. It tips. At the edge of the shelf, where the flat pale sediment of relatively shallow water gives way to the first long descending slope, the geometry of the ocean changes entirely. The bottom drops away, not like a cliff falling sheer and sudden, but like the far edge of a vast tilted plane, angling downward into water that grows darker and colder as the slope continues. Along this rim, where the descent begins, where shelf becomes slope and the bottom moves away from any proximity to sunlight, the conditions for a particular kind of life become, in some stretches, very nearly perfect. The currents that ride along the shelf edge accelerate where they meet the changing contours of the seafloor. Water presses against the rising lip of the shelf and moves faster, more insistently, carrying particles with it. And in that accelerated flow, along hundreds of kilometers of descending rock and hard substrate, cold water coral reefs have been growing in total darkness for longer than any human civilization has existed. The shape of the seafloor matters more to these reefs than almost anything else. Flat, featureless sediment offers no foothold, no surface for a polyp to settle and begin its slow calcification. What cold water corals require is hard rock, the kind that resists burial, the kind that juts from a slope or a ridge and stands above the surrounding accumulation of fine sediment. Shelf edges provide this. The structural relief of the continental margin, its outcrops, its fault scarps, its exposed carbonate platforms, offers the bare, firm surface that a settling coral larva needs. Once a single polyp takes hold and begins building its small calcium carbonate cup, others follow. The branching colony expands. Over decades, the colony becomes many colonies, each one adding its skeleton to an interlocking three-dimensional framework that rises slowly from the rock. And because the current that flows along the shelf edge carries food, the slow rain of organic particles drifting down from far above, the reef, once established, can sustain itself indefinitely in the dark. The Norwegian shelf edge is one of the longest stretches of confirmed cold water coral reef habitat on Earth. Along the margin of the Norwegian Sea, from the waters north of Bergen to the stretches approaching the Lofoten Islands, reef systems have been mapped running for tens of kilometers in continuous or near continuous bands. These are not scattered colonies or isolated patches. They are reef systems in the full ecological sense, dense, structurally complex accumulations of branching calcium carbonate, built by the coral Lophelia pertusa, extending across sections of hard seafloor that in some places measure hundreds of meters across. The water here is cold, running between 4 and 8 degrees Celsius at typical reef depths. It is dark, completely and permanently, with no seasonal variation in the quality of light because there is no light of any kind. The pressure at 200 to 400 meters is enough to compress the air in a human lung to a small fraction of its surface volume, and in these conditions, the reef grows. Rockall Bank lies further west, out in the open North Atlantic, a submerged plateau rising to within a few hundred meters of the surface and surrounded by very deep water on all sides. The Rockall Bank itself is geologically ancient, a fragment of continental crust that separated from Europe as the Atlantic opened, and its flanks drop away steeply into the surrounding ocean basin. Along these slopes, particularly in the transition zone between 400 and 800 meters, cold water coral reefs have established themselves on the harder substrate of the bank's flanks. The currents that move around Rockall are strong and persistent, driven partly by the bank's own obstruction of the general North Atlantic circulation. Water flows up and over the bank's shallower sections and swirls along its margins, and this constant motion delivers suspended food particles to the reef communities along the slopes. The reefs here are far from any coast, far from any regular human observation. They have been growing in total darkness in the middle of the open ocean. Between these two areas, and extending southward toward the British Isles, the Rockall Trough cuts through the seafloor like a long basin, thousands of meters deep at its center, its margins sloping upward on either side toward shallower ground. It is along one section of this trough's floor, at around 1,000 meters depth, that a field of coral structures called the Darwin Mounds occupies a stretch of sediment that at first appears, to a camera descending from above, simply flat. The trough floor at this depth is sandy and even. There are pockmarks in the sediment, shallow depressions formed by gas or fluid moving up through the sediment layer, and scattered across this pockmarked plain, rising a few meters from the surrounding flat, are hundreds of individual mounds. Each one is capped with living Lophelia pertusa, Each one is built from the accumulated skeleton of past colonies, coral growing on coral, the living surface always a thin veneer over the older material beneath. The mounds are not tall, 5 meters is significant here, but across the width of the field, the scale of the accumulation becomes apparent. This is not incidental growth. This is a landscape with its own topography, its own internal structure, its own community of organisms living within and between the mounds. At 1,000 meters, the pressure at the Darwin Mounds reaches approximately 100 atmospheres, 100 times the weight of air pressing against every surface at sea level. The temperature here is close to 6 degrees Celsius, cold enough that the metabolic pace of most warm water organisms would slow towards stillness. There is no light, not dim light, not the faint blue filtering of very shallow water. Absolute dark, without qualification, the kind of dark in which the concept of color becomes meaningless because there is nothing to create distinction. The sandy floor between the mounds absorbs whatever movement comes through it without reflection. The coral structures themselves, pale and branching, are invisible to any eye in the absence of illumination. Only the current moving through them exists as sensation, the slow, persistent pressure of water carrying its cargo of fine particles and small organisms across the structure of the reef. Far from the Darwin Mounds, off the southwestern coast of Ireland, where the Celtic margin descends into deeper water, the seafloor tells a different story. Here, in the Rockall Trough, and along the margins of the Porcupine Sea Bight, a large indentation in the Irish continental margin where the shelf curves inland. Before dropping away, some of the largest deep-sea coral structures in the world have accumulated. The Belgica Mound province occupies a section of the Irish margin where, for reasons that combine the specific geometry of the seafloor, the nature of the local current system, and the long availability of hard substrate, cold water coral has been growing for an extraordinarily long time. The mounds here are not 5 meters tall. Some of them rise more than 100 meters from the surrounding seafloor. Their bases span several kilometers. More than 60 individual mounds have been mapped in this province, and they cover a stretch of the Irish margin that is wider from one side to the other than many surface cities. Standing at the base of one of these mounds, in the conceptual sense of standing, in the imaginative sense of being present at the seafloor and looking upward, the mound rises through the dark water in a way that has no surface equivalent. The slope is gradual in some sections, steeper in others, where the accumulated framework is more dense. The flanks are colonized by sponges and new coral recruits, organisms that have found purchase on the outer surface of older carbonate. Higher up, where the mound narrows toward its summit, the living coral is more dense, the framework more intact, the polyps more actively extended into the current that flows across the mound's upper reaches. The summit is alive, the flanks are alive in places and quiet in others, and the base of the mound, buried in accumulated sediment, is mostly dead framework, the calcium carbonate skeletons of colonies that lived and grew and died hundreds or thousands of years ago, their contribution to the mound now incorporated into its physical foundation. The whole structure is both cemetery and living reef at once. The dead and the living occupy the same body of stone. The seamount is a different kind of place, where the coral mound grows from the seafloor and rises within the broader context of a slope or a basin. The seamount is itself a mountain, a volcanic peak rising from the deep oceanic crust, often from depths of thousands of meters up toward the surface. Some seamounts breach the surface and become islands Most do not, their summits resting hundreds of meters below the waterline, in a depth zone that shares some characteristics with the shelf edge, but adds the particular enhancement of a peak projecting into water that moves faster the more it is constricted and accelerated by the obstacle below. Water flowing past a seamount cannot go through it. It must go over or around it, and in doing so it accelerates, compresses, and concentrates whatever it carries. For cold water coral, this concentration of current means a concentration of food. Seamounts in the North Atlantic, the Pacific, the Southern Ocean, and the Indian Ocean all host cold water coral communities on their slopes and at their summits, wherever the substrate is hard and the current strong enough to prevent sediment from burying the reef. The physical experience of a seamount summit, translated through the qualities of the water that moves across it, is one of persistent, forceful flow. At 200 to 400 meters below the surface on a typical North Atlantic seamount, the current across the peak can run at speeds that would feel to any structure in its path like a strong, steady wind. Sediment does not accumulate here. The flow keeps the rock clean, bare, exposed, exactly the surface a coral larva needs when it settles. Fine organic particles that might drift slowly downward through still water are kept in suspension by the current, circulating repeatedly across the reef surface and increasing the likelihood that a waiting polyp will intercept them. The seamount, through its simple geometry of projection and constriction, acts as a gathering point for whatever the surrounding water carries. Norwegian fjords offer yet another variation on the same theme. The fjords cut deep into the Norwegian coast, long, narrow, steep-sided arms of the sea that in some cases reach back inland for many tens of miles and down to depths of hundreds of meters. The surface water in a fjord is often relatively fresh, diluted by the rivers and runoff that drain from the surrounding landscape. But deeper below the surface layer, cold, dense Atlantic water flows along the fjord floor. This cold, dense water is saltier and heavier, and it moves inward from the open sea along the bottom of the fjord channel, displacing lighter surface water upward. In the deeper sections of fjords, like Korsfjord, Trondheimsfjord, and Sognefjord, this cold, dense bottom current creates conditions that are chemically and physically very similar to the shelf edge environment at several hundred meters depth, but at depths of only 30 to 40 meters below the surface. 30 meters in open coastal water that is well within the photic zone. Light reaches 30 meters in clear tropical water with enough intensity to drive productive photosynthesis. But a Norwegian fjord at 30 meters depth in the cold, dense water flowing along the floor of a steep-sided channel is not a sunny, shallow water environment. The light that penetrates the fjord surface is scattered and attenuated by the optical properties of the water column. The cold, nutrient-rich bottom layer is poorly lit at best, and in the deeper fjord channels, where the angle of the terrain and the optical depth of the water combine, essentially dark. And in this unexpected near-surface darkness, Lophelia pertusa reefs grow within what would, in any other context, be considered the shallow coastal zone. These are the most accessible cold water coral reefs in the world, encountered not at the far end of a multi-day research expedition to deep water, but relatively close to shore in fjords that have been navigated by fishing boats for centuries. The fjord reefs also illustrate something that the shelf edge and seamount reefs share. The logic of what makes cold water coral habitat is not primarily a logic of depth. Depth is a proxy for the conditions that cold water coral actually requires: darkness, cold temperature, food-laden current, and freedom from the competition of light-dependent organisms like algae and tropical coral species. In a fjord where cold, dense water creates those conditions at 30 meters, the reef is at 30 meters. In the open North Atlantic, where those conditions do not appear until 600 meters, the reef is at 600 meters. The organism is not seeking a specific depth. It is settling where the conditions are met, and the conditions can be met at a wide range of depths across different parts of the ocean. The pressure at those different depths varies considerably. At 30 meters in a fjord, the pressure is modest, about 3 atmospheres, something that any recreational diver encounters without difficulty. At 500 meters on a shelf edge, it is closer to 50 atmospheres, well beyond any human diving capacity. At 1,000 meters, at the Darwin Mounds, it is 100 atmospheres. At 2,000 meters, on the deeper slopes where cold water coral communities have also been documented, the pressure exceeds 200 atmospheres, which is a number that is difficult to make physically intuitive. Consider a surface roughly the size of a thumbnail. At 2,000 meters depth, the weight of water pressing on that thumbnail-sized surface is equivalent to the weight of a small car. Every surface in the body of any organism living at that depth experiences that force simultaneously from every direction at once, from above, from below, from all sides, so evenly distributed that organisms adapted to it do not feel it as a crushing force, but simply as the baseline condition of their existence. Cold water coral polyps have no physiological structure oriented toward light. There are no photoreceptors of the kind that corals in the photic zone possess, no molecular machinery tuned to detect the quality or intensity of sunlight, no symbiotic partnerships that require light to function. The architecture of these organisms is oriented entirely in other directions, toward the current, toward the chemical signals that indicate the presence of food, toward the substrate below and the water above that delivers what they need. Pressure, in the biochemical sense, has shaped the lipid composition of their cell membranes and the proteins that carry out cellular functions, such that these biochemical processes operate normally at pressures that would denature the equivalent proteins in a surface organism. The deep cold is not something these corals tolerate in spite of themselves. It is the medium they are built for. The geography of cold water coral reefs is ultimately a geography of current and topography. Wherever the seafloor rises or tilts in a way that accelerates water movement, wherever the currents carry enough organic material to sustain suspension-feeding communities, wherever hard substrate is exposed and free of smothering sediment, the reef can establish. This pattern repeats across ocean basins with the consistency of a physical law. The same branching white framework, built by the same biological logic, appears along the edge of the Rockall Bank and along the Norwegian shelf, along the Irish margin, and along seamounts in the open Atlantic, inside Norwegian fjords, and across the floor of the Porcupine sea bight. It appears in the Pacific, in the Gulf of Mexico, around New Zealand, in the Mediterranean. The environmental template is not unique to any one ocean. It is a template written in the physics of flowing water and hard rock, and where the template occurs, the reef follows. Off the northern coast of Scotland, the shelf edge descends through water that is cold and runs with persistent tidal currents, driving along the Atlantic face of the British margin. Here, too, surveys have documented cold water coral communities on the rock outcrops that line the descending edge of the shelf. The specific geometry of this margin, the way the shelf narrows and the slope steepens, the way the tidal currents are amplified by the topographic constriction of the waters between Scotland and Iceland, creates along slope velocities high enough to maintain the reef in conditions of reliable food supply. These reefs are not mapped as extensively as those off Norway or Ireland, in part because the logistics of deep water survey work are demanding, and these particular areas have received less systematic attention. What surveys have been conducted find the same structures, the same branching framework, the same pale architecture extending across stretches of exposed rock in the permanent dark. The Mediterranean might seem an unlikely home for cold water coral. The Mediterranean is warm at the surface, and deep water coral is most commonly associated with very cold oceanic conditions. But the deeper waters of the Mediterranean are cold enough, and in certain regions, particularly along the margins of the western Mediterranean basin and around the Strait of Sicily, surveys have found cold water coral communities at depths between 200 and 500 meters, on water that is colder than the surface, but not as cold as North Atlantic deep water. The conditions here are in some ways marginal for the species. The Mediterranean is nearly enclosed, and its deep circulation is slower and more constrained than the open ocean. Aragonite saturation levels, which influence how easily the coral can secrete its calcium carbonate skeleton, are adequate but not as elevated as in the North Atlantic. The reefs that establish here are in some cases less structurally developed than their Atlantic counterparts, and yet they are present, distributed across sections of the Mediterranean seafloor in habitats that confirm the breadth of the environmental template that this organism can inhabit. New Zealand sits at the center of a section of the Pacific with an unusually complex seafloor topography, ridges, rises, seamounts, and shelf margins created by the meeting of the Pacific and Australian tectonic plates. The deep waters around New Zealand are cold and well-aginated, with currents shaped by the influence of the Southern Ocean on the broader Pacific circulation. Cold water coral communities have been documented here on the flanks of the Chatham Rise, on seamounts throughout the New Zealand exclusive economic zone, and along shelf margins on both the eastern and western coasts of the island system. These Pacific cold water coral communities are less well studied than their Atlantic counterparts, but the organisms found there are in many cases closely related or taxonomically identical to those found in the Atlantic, a fact that reflects both the global connectivity of deep ocean larval dispersal and the global consistency of the conditions that support cold water coral growth. The full extent of cold water coral distribution is a number that continues to be revised. Each major survey program that has used remotely operated vehicles to examine deep water habitats in previously unsurveyed areas has found more reef than the estimates that preceded the survey assumed. The conservative estimate, based on confirmed documented locations, places the global area of cold water coral reef at figures comparable to the total area of tropical coral reef worldwide. Less conservative estimates, accounting for the large proportion of suitable habitat that has not yet been examined, suggest that cold water coral reef may cover considerably more seafloor than all tropical reef systems combined. Given that the deep ocean floor represents the largest continuous habitat on Earth, and given that relatively little of it has been observed at close range, both estimates carry substantial uncertainty. What is not uncertain is the distribution. Every major ocean basin, every major continental margin that has been surveyed at appropriate depths, every seamount field that has been examined has returned evidence of cold water coral. The weight of water above all of these places is enormous, not in the way that weight is experienced at the surface, where it falls in one direction and can be resisted and braced against, but in the encompassing way of pressure at depth, where it arrives from everywhere simultaneously and has no preferred direction. At 100 meters depth, the pressure is 10 atmospheres. At 500 meters, 50. At 1,000 meters, 100. At 2,000 meters, the entire column of seawater above presses inward on every square inch of every surface with the same patient, total force. The corals that grow across these ranges of depth are not aware of this in any meaningful sense. There is nothing in their physiology analogous to discomfort or endurance. The pressure is simply the medium. The temperature is simply the temperature. The dark is simply the condition of the water they inhabit, the current they feed from, the substrate they have calcified themselves to, across the full span of their lives. The reef in the dark is not smaller for the absence of light. The branching framework of a cold water coral colony does not grow less elaborate because there are no photons to illuminate it. The community of organisms that shelters within and around the reef framework is not thinner because the water above is black rather than blue. What the dark creates is not a diminishment. It creates a particular set of conditions, cold, pressurized, current-swept, chemically specific, and within those conditions, the reef is exactly as complete as any structure can be. The mound rises, the polyps extend their tentacles. The current delivers its cargo, the framework accumulates, year by year and decade by decade, in the total stillness of the permanent night. And in that stillness, the pressure of the ocean above settles into the rock of the mound, and the mound does not move. It persists in the cold dark with the same indifference to disturbance that the deep ocean has always maintained. 200 atmospheres, or 30 meters below the surface of a Norwegian fjord, or 1,000 meters below the surface of the open Atlantic. The reef, wherever these conditions combine, is there, building, still, patient in the way that only something without awareness of time can be truly patient. The water moves through the branches hold, the darkness holds around them, and the weight of all that water, pressing in from every direction with perfect evenness, becomes simply the silence the reef rests inside. A single particle is falling. It is small enough that the word small does not quite reach it. A few millimeters across at most, and irregular in shape, built from fragments of things that were once alive. A shred of algae cell wall, a curl of shed mucus, a fecal pellet no larger than a grain of salt, all of it bound loosely together by the chemistry of seawater into something that drifts rather than drops. It left the surface perhaps a week ago, perhaps two. The sunlit water where it formed is now 800 feet above it, then 1,000, then more, and still it descends, slower than smoke, slower than dust, settling in a still room, drawn by gravity, in increments so small that the falling is nearly indistinguishable from hovering. The water around it is black. There is no light here of any kind, not the faintest filtered remnant of the surface day, not a bioluminescent flash from any passing animal. The darkness is total, and has been total for as long as any particle has been falling through this column of water. And still the particle falls. Below it, though it has no way to know this, a reef is waiting. The reef does not wait in any conscious sense. It is simply there, as it has been there for a very long time, built from calcium carbonate, and cold in the patient geometry of colonial animals that have never needed anything from the sun. The particle will take several more hours to reach it, and when it does, it will be caught. This is how a reef in permanent darkness eats. The process begins, as most processes do, far from where its consequences are felt. High in the ocean, in the bright shallow band where sunlight still penetrates, tiny plant cells drift in their billions, producing carbon through photosynthesis, dividing and multiplying through the long days of spring and summer, building the living mass of the phytoplankton bloom that turns whole stretches of surface water a faint greenish blue when seen from above. These cells live and divide, and are eaten and die, and are eaten and die, and what they leave behind, their own dead matter, the waste of the animals that ate them, mucus shed from grazers moving through the bloom, transparent feeding webs cast out and abandoned by tiny drifting creatures. All of this begins to sink, not quickly, not in any way that would register as falling to a human eye, but steadily and without stop. For as long as the ocean has been an ocean, organic material has been drifting downward from the lit surface into the dark. Scientists call it marine snow, and the name is accurate in the way the best names are. It captures the scale and the motion and the quality of the thing without explaining it, and it stays in the mind like a picture. Marine snow falls through the whole water column, from the surface down through the twilight zone where the last traces of sunlight fade, through the fully dark middle waters and on toward the floor. It moves at different rates depending on the density of individual particles and the turbulence of the water around them. Some particles aggregate as they fall, sticking to one another, growing heavier, sinking faster. Others are intercepted by animals, copepods, and small crustaceans moving through the water column, feeding on the organic rain as it passes, incorporating the carbon into their own bodies before releasing it, again as waste that sinks in turn. The marine snow that reaches the deep sea floor is not the same material that left the surface. It is transformed, eaten and excreted, and repackaged, reduced in volume, enriched in some compounds, and depleted in others. But it arrives, across the whole deep ocean, across every basin, across every kilometer of dark seafloor. The slow rain of organic matter from the surface is constant. Nothing living in the deep is entirely disconnected from it. The coral polyp is small, pulled back into its calcium carbonate cup. It is a few millimeters in diameter, a soft cylinder of tissue sitting in a hard white socket of its own making. Extended, it becomes something else, a ring of tentacles reaching outward from a central mouth. Each tentacle armed along its length with nematocysts, small cellular capsules that can discharge a coiled thread tipped with paralyzing chemistry at the moment of contact. The tentacles move in the current, not with intention, but with the slow passive responsiveness of a structure shaped by water over long time. They extend when conditions are right and retract when they are not. On the reef, in the dark, the current is moving. The water arrives along the slope of the continental shelf edge, drawn by tidal forces and the circulation patterns of the deep basin, and as it meets the topographic relief of the reef structure, the branching framework of interlocked coral colonies, rising several meters from the surrounding rock, it accelerates. The boundary between still water and moving water shifts. Eddies form and dissolve in the lee of coral branches. Particles suspended in the current. Marine snow aggregates and small living animals carried along in the flow are delivered at a rate that varies with the speed and direction of the water, and the variation is not random. There are pulses. The tide rises and the current picks up, and across the surface of the reef, polyp by polyp, across thousands of individual colonies, the tentacles extend. This is not coordination in any willed sense. Each polyp extends when the hydrodynamic stimulus is sufficient, when the pressure and chemistry of moving water tips a threshold in the tissue's sensory cells. But the result at the scale of the whole reef is a kind of synchrony, a surface that was still becoming a surface that is reaching the entire living face of the reef, activated by a common cause, the water moving through and among them all at once. A marine snow particle contacts a tentacle. The nematocysts discharge, The thread penetrates the particle and holds it, and the tentacle begins to curve inward toward the polyp's mouth. The particle, this small aggregated fragment of dead algae and shed mucus and compressed waste that left the surface weeks ago and fell through 800 meters of dark water to arrive here, is drawn through the mouth and into the gastrovascular cavity of the polyp, where digestion begins. The carbon and nitrogen and phosphorus that were once inside a living phytoplankton cell are now inside a living coral cell incorporated into tissue, building the polyp's own proteins and the calcium carbonate of its skeleton. The reef grows. The same transaction is happening in thousands of places across the reef simultaneously, each polyp feeding in the dark at its own rate, each one contributing to the slow thickening of the structure that surrounds and supports it. Not every tentacle makes contact in any given current pulse. Many particles drift past without touching. The process is not efficient in any engineered sense, but efficiency is not the frame that applies here. The frame is time. Given enough time and enough current and enough marine snow falling from above, the reef feeds itself and grows and persists. Marine snow is not the only food. The water moving across the reef also carries animals, small live zooplankton, copepods and amphipods, and tiny crustaceans moving through the dark water column on their own trajectories. A copepod is different from a marine snow particle in all the ways that matter to a coral polyp. It moves, it changes direction, it carries a higher concentration of the nutrients the polyp most needs, nitrogen in particular, bound in proteins, and lipids and forms the polyp's tissue can use directly. To catch a copepod requires more from the tentacle than to catch a particle of marine snow. The nematocysts must discharge accurately, and the copepod's escape reflex, the rapid backward kick that is among the fastest accelerations in the animal world, must be overcome before contact is broken. Many escape, some do not. A copepod moving through the space between two coral branches in the dark makes contact with a tentacle tip. The nematocysts fire. The coiled threads extend and penetrate the copepod's cuticle and deliver their chemistry, and the animal's body goes still, not dead, but paralyzed. The nervous system interrupted. The tentacle curves. Adjacent tentacles respond to the presence of the prey, bending toward it, helping to secure it. The copepod is moved toward the central mouth and drawn inside. The digestion of a live animal is different from the digestion of a marine snow particle. The polyp extracts a richer and more varied chemistry from a copepod's body: amino acids, fatty acids, trace elements bound in muscle and nervous tissue, all of them becoming, in time, part of the polyp itself. The reef is fed by both, by the slow continuous rain of dead organic matter drifting down from a surface it will never know, and by the animals moving through the dark water around it, alive until the moment they are caught. The two food sources are not in competition. They are complementary, the marine snow providing a baseline of carbon and nitrogen that arrives regardless of season or condition, the live prey providing richer packages of nutrition that the reef intercepts when the current delivers them. Between the coral branches, in the small enclosed spaces created by the geometry of the framework, the current slows. Particles that the current would carry past in open water settle here, drifting into recesses where the flow is gentle. The reef creates its own microenvironments, pockets of relative calm inside a structure built by organisms feeding on turbulence. In these pockets, other organisms have settled, sponges and brittle stars, and small worms, each one feeding in its own way on the same rain of particles, each one adding its own organic waste to the local chemistry. each one contributing in small ways to the cycling of nutrients within the reef structure. The reef feeds them and they feed the reef's chemistry, and the whole enterprise turns slowly in the dark, driven by nothing except the movement of water and the fall of matter from above. The current does not blow constantly, it pulses, it strengthens and weakens with the tides, with the passage of storms at the surface far above, with the seasonal shifts in the density structure of the water column that alter how deep-reaching currents flow. The reef experiences these variations not as weather, but as changes in the rate and composition of food delivery. More particles when the current is strong, fewer when the water is still, different particles in different seasons, the whole chemistry of what arrives, changing slowly through the year. In summer, the surface above is warm and stratified. The sun heats the top layer of the ocean, and the density difference between warm surface water and cold deep water strengthens, slowing the vertical exchange of materials between the lit surface and the dark deep. Less organic material reaches the reef in midsummer because the stratification that drives the spring bloom also tends to slow the flux of particles downward once the bloom has run its course. The reef continues to feed on what the current brings, but the supply is reduced and the polyps draw more on the reserves stored in their own tissue from earlier months. Then autumn begins to come. At the surface, 800 meters above, the water cools. The temperature gradient between surface and depth weakens. The stratification that kept the water layers separate begins to break down, and as it does, the upper ocean mixes more freely. Nutrient-rich water from below rises to meet the cooling surface, and if conditions are right, and in the North Atlantic in autumn they often are, a second, smaller bloom begins. Phytoplankton grow again in the cooling autumn light, not as intensely as the spring bloom, but substantially, adding another pulse of organic production to the surface layer before winter shuts the process down entirely. The dead cells and particles from this autumn production begin their descent. They fall through the same path as the spring bloom's aftermath, through the twilight zone and into the dark, but they travel a subtly different chemistry. Cells that grew in cooler, more turbulent conditions, aggregated differently, carrying different ratios of carbon to nitrogen, and they travel in greater numbers than the slow summer flux. The flux of particulate organic carbon reaching the deep reef roughly doubles in the weeks following the autumn bloom, a measurable pulse that arrives at depth perhaps weeks after the bloom itself has already ended at the surface. The reef responds, not with any sudden change, nothing here is sudden, but with a measurable shift in the behavior of the polyps. Extended tentacles are held out longer into the current. Feeding rates, measured by the rate at which particles are intercepted and consumed, increase. In the weeks that follow the autumn organic pulse, cold water coral colonies show higher rates of calcification, adding slightly more to their skeletons than they do in the slower summer months. Reproductive activity increases as well. The polyps, fed more richly, invest in their own reproduction, releasing gametes into the current to drift and potentially settle on new surfaces of bare rock. The reef does not know it is responding to an autumn bloom. There is no knowledge here, no perception of seasons, no connection between the polyps feeding in the dark and the cooling surface light hundreds of meters above. There is only the chemistry of what arrives, the ratio of organic carbon to inorganic matter in the particles touching the tentacles, the concentration of amino acids in the prey items caught from the current, and the biological responses that specific chemistries trigger in coral tissue that has been shaped by long time to respond to exactly these signals. The spring bloom at the surface and the autumn bloom at the surface and the long, slow summer flux between them are experienced by the reef as a slow rhythm of abundance and reduction and abundance again, the chemical echo of a cycle driven by light. The reef has never received and never will. The light changes the surface changes the flux changes the reef. By the time any given photon of spring sunlight has completed its effect on the reef below, through the bloom it drives, through the cells the bloom produces, through the particles those cells become, through the fall of those particles, through 800 meters of dark water, through the digestion of those particles by polyps extending their tentacles in the dark. Months have passed, perhaps half a year. The sunlight of April reaches the reef in October, broken apart and reassembled into 100 intermediate forms, arriving not as light, but as chemistry, as carbon in a particle, as nitrogen in an amino acid, as phosphorus dissolved from a dead cell's membrane. There is in some places another source. In the Gulf of Mexico, where cold water coral colonies grow in deep water as they do across the Atlantic, the seafloor is not uniform. It is threaded with fault lines and sediment structures through which pressurized gases and fluids from below move upward, seeping through cracks in the sediment and releasing methane and other reduced chemical compounds into the water column above. These cold seeps, as they are called, support dense communities of microorganisms that use the chemical energy in methane and hydrogen sulfide to build organic carbon, not through photosynthesis, not using sunlight at all, but through chemosynthesis, a parallel biological logic that runs on the chemistry of the deep earth rather than the light of the sun. The Lophelia pertusa colonies growing near these seeps feed in the same way as colonies anywhere else, extending tentacles into the current, capturing particles and prey. But the chemistry of the particles near a cold seep is not quite the same as the chemistry of particles far from one. Some fraction of the organic material in the water column above a seep carries the chemical signature of chemosynthetic production, carbon atoms with a subtly different ratio of isotopes than carbon derived from surface photosynthesis. And in the tissue of cold water coral colonies growing near Gulf of Mexico seeps, this signature has been detected. Not as the dominant carbon source, the coral is still primarily fed by the same marine snow as everywhere else, but as a measurable thread, a minor chemical record woven into the polyp's tissue, indicating that a small fraction of what built this colony came not from the surface, but from the earth beneath it. The deep reef in these places is connected not only upward to the sunlit surface, but also downward, in a faint and indirect way, to the chemical energy stored in the rock and sediment of the deep seafloor itself. The coral does not seek it out. The chemistry arrives in the current, indistinguishable to the polyp's tentacles from any other particle, and is incorporated without any awareness of its origin. The isotopic record is preserved in the skeleton, waiting to be read by a scientist dissolving a few milligrams of carbonate into acid in a laboratory on the surface, centuries or millennia later. The current shifts again. The water that has been moving along the reef face from one direction begins to slow as the tidal cycle turns, and for a time the flow is minimal, the delivery of particles reduced, the polyps partially retracted into their cups. In the stillness between tidal pulses, the reef is a different structure, less animated, less actively engaged with its environment. The surface that was reaching, now resting, drawing the small motion of its tentacles back inward, waiting for the water to move again. Waiting is not the right word, but there is no word for what an organism without a nervous system does in the interval between feeding events. The polyps simply exist, metabolizing at the rate that the temperature of the water permits, slowly, because the water is cold, because the temperature here is somewhere between 4 and 10 degrees Celsius, and at these temperatures, the biochemical reactions inside the cells run at a fraction of the speed they would at tropical reef temperatures. The cold slows everything. It slows growth, it slows feeding, it slows reproduction, and it slows death, in the sense that the same chemical reactions that drive aging and cellular degradation also slow in the cold, and a cold water coral polyp may live for decades, its colony for centuries, its reef for timescales that have no good comparison in the warm water world. The water begins to move again. A tidal current resumes, not as dramatic as the strongest pulses, but sufficient. Tentacles extend. The surface of the reef becomes active again, each polyp responding to the same stimulus, the whole living face of the structure, turning outward into the current as it thickens with moving water. A marine snow particle, larger than most, a dense aggregate of algae fragments and mucus and the shed outer layer of a copod from somewhere in the middle water column, drifts sideways in the current and contacts a coral branch not quite at a polyp, but at the dead skeleton, between two living cups. It sticks briefly, then dislodges, then drifts again, moving along the branch until it reaches the edge of a living polyp's reach. The tentacle extends slightly further in response to the chemical signal the particle carries. Amino acids dissolved in the boundary water, the faintest gradient of organic chemistry detectable by the tissue, and the nematocyst's fire. The particle is held. The tentacle curves. The particle moves toward the mouth. This transaction has been happening in one form or another for as long as this colony has existed. The colony is not young. Its oldest branches are decades old at minimum, possibly much older, built from calcium carbonate laid down by polyps that are now dead, their tissue gone, their skeletons incorporated into the branching framework that the living polyps sit upon today. The framework itself is the accumulation of all those earlier feeding events, all those particles captured, all that carbon transformed into carbonate by the chemistry of calcification, all that slow growth added up over time into a structure several meters across. Every branch is a record of past feeding. Every cup holds a living polyp engaged in the same feeding that built the branch beneath it. The particle is digested. The carbon in it enters the polyp's metabolic cycle. Some of it is burned for energy. Some of it is incorporated into new tissue, building the cytoplasm and proteins of the living cell. And some of it, combined with calcium dissolved from the surrounding seawater and carbonate ions produced by the polyp's own metabolic activity, is deposited as calcium carbonate at the base of the polyp's cup, thickening the wall by a fraction of a millimeter, adding to the skeleton's mass by an amount too small to measure by feel, but real and cumulative and persistent. The reef grows by these fractions. Not quickly, not in any way that registers as change on a human timescale of observation, but steadily and without stop, as long as the current brings food and the water holds the chemistry that calcification requires. The fraction of a millimeter added tonight will still be there in 100 years, in 1,000 years, in the sediment core that a future instrument pulls from the mound's interior to read what the ocean was doing in this century. The carbon that was marine snow falling from a spring bloom on the surface, captured by a tentacle in the dark, transformed into carbonate by the chemistry of a small cylinder of tissue deposited as skeleton. That carbon will outlast everything that produced it. The current carries another pulse of water along the reef face. The water is not different from the water a moment before in any way that would be visible, but its chemistry has shifted slightly. The organic load fractionally higher, the concentration of small particles sufficient to make contact with the outstretched tentacles more frequent. Feeding rates across the reef surface increase slightly. The whole slow transaction continues. Far above it is night. Not the permanent night of the deep reef, but the ordinary night of the ocean surface, the rotation of the earth having carried this part of the Atlantic into darkness for the hours between dusk and dawn. In the surface water, zooplankton that spent the daylight hours in deeper, darker water have migrated upward to feed in the rich surface layer under the cover of darkness, and by dawn they will descend again, carrying the carbon they consumed upward during their feeding back down into the middle depths when they retreat. This vertical migration, one of the great daily movements of carbon in the ocean, shifts organic matter deeper into the water column than sinking particles alone would carry it, and some of the zooplankton that migrate downward will never reach the surface again, caught by predators in the deep, or dying there and sinking further. Their carbon joins the general downward flux, adding to the marine snow that will, weeks or months from now, reach the reef below. The surface night does not reach the reef. The reef has no night and no day, no cycle tied to the rotation of the planet, no rhythm organized around the sun. What it has instead are the tidal rhythms, the 12-hour cycles of current strength that carry more food in one half of the tidal period than the other, and the longer seasonal rhythms of flux from above, and the irregular rhythms of storms and circulation shifts that alter the deep current patterns over weeks and months. These are the reef's equivalent of time, not sunrises and sunsets, not the warmth and light of day contrasting with the cool dark of night, but current and calm, pulse and stillness, the arrival of organic chemistry in varying concentrations across the hours and months and years. The particle that fell from the surface weeks ago, that was caught by a tentacle and drawn through a polyp's mouth, and digested and incorporated into tissue, and partly deposited as calcium carbonate in the wall of the polyp's cup. That carbon is now part of the reef. It will remain part of the reef as long as the skeleton persists, which may be centuries. Long after the polyp that deposited it has died and its soft tissue decayed, the carbonate will remain, locked in the white branching framework, covered eventually by new coral growing above it, buried in the slow accumulation of the mound. The particle's journey is over. It fell from the surface as marine snow, drifted downward through several hundred meters of dark water, was captured in total darkness by a living organism it had no awareness of, and became, through the chemistry of a small soft animal's body, a permanent part of the reef. This is how the reef is built, not by light, not by any energy that comes from above in the form of photons striking photosynthetic cells, but by the chemistry of what falls and what swims and what the current delivers, captured particle by particle, prey item by prey item, across timescales that belong to geology rather than to any individual life. The reef is the record of all those transactions. Every branch is a column of captured and converted organic matter. Every cup that holds a living polyp tonight was built by the feeding of past polyps on past particles from past surface blooms that occurred before any living human was born. The current pulses again. The tentacles extend. Somewhere in the water above the reef, a small crustacean is moving through the dark, navigating by the chemical gradients that signal the presence of food and predator, completely unaware of the branching structure rising beneath it from the seafloor. It will reach the depth of the reef canopy in a few minutes, carried partly by the current and partly by its own swimming, and when it contacts a coral branch and a tentacle reaches toward it, it will react with the explosive escape response that has worked for countless generations of its kind. Whether that response succeeds on this particular night against this particular tentacle is a matter of contact geometry and nematocyst timing and the precise position of the animal's body relative to the discharging cells. The reef does not know the outcome. It reaches into the current with the same patient extension of tissue that it has always offered, and the current brings what it brings, and the reef feeds on what the tentacles hold. The organic pulse from the autumn bloom is still arriving, still measurably elevated above the baseline summer flux. The polyps have been feeding at slightly higher rates for several weeks, and the calcification record in their skeletons, if measured now, would show a faint thickening of the walls laid down in these weeks compared to the walls laid down in July and August. This thicker layer will be visible, with the right tools and lighting, in a cross-section of the skeleton, years from now, a pale band in the carbonate, slightly denser than those on either side, recording these particular weeks, this particular autumn pulse, this particular season of organic abundance, drifting down from a bloom that lit the surface for a few weeks and then was gone. The bloom is already gone. At the surface, the autumn production peaked weeks ago, and now the cooling water and the shortening days have slowed photosynthesis to near nothing. The surface is dark and cold, and the bloom's cells have sunk, but their chemistry is still arriving at the reef below, still feeding the polyps, still being incorporated into tentacles and mouths and calcium carbonate walls in the permanent darkness of the deep. The surface responded to autumn light. The reef is responding now. Between the two responsing, now, months have passed, and in those months the carbon has moved through a dozen different forms: living cell, dead cell, marine snow particle, tentacle contact, gastrovascular chemistry, deposited carbonate, each transformation one step further from the original photon that drove the whole sequence. The spring that follows will send another pulse. The summer will send a reduced one. The autumn will send another. And the reef will feed on all of them, slowly, in the dark, adding fractions of millimeters to its skeleton with each passing season, building the mound higher by increments too small to feel, but too consistent to stop. A marine snow particle drifts against a tentacle. The nematocysts fire. The tentacle curves inward. The particle is drawn through the mouth into the dark interior of the polyp, where digestion begins, and the slow translation of surface light into deep stone continues, quietly, in the absolute dark, as it has continued for thousands of years, and will continue for thousands more. The skeleton came first. Long before the living animal, there was the calcium carbonate shell of something that had already finished growing, already finished feeding, already finished whatever slow commerce it conducted in the dark. A single branch of dead Lophelia framework, pale and branching and intricate as frost on glass, sits wedged between two outcrops of bare rock 400 feet below the surface of the ocean. No light reaches it. No warmth from the sun has ever touched this particular stone, and yet pressed into the inner angles of the branch, tucked between two ribs of carbonate, where the geometry of the dead coral creates a space just wide enough. Something is living there, something small and many-legged and perfectly still, resting against the skeleton of an animal that died perhaps a century before it was born. This is how the reef begins, in a sense, not with growth, but with inheritance. The dead framework persists. It provides what the living coral cannot yet provide for itself: a hard surface, a sheltered cavity, a geometric complexity that breaks the current into gentler eddies and creates in a landscape of otherwise bare and exposed rock something more like a home. A single dead branch, cleaned of its tissue, is already a small architecture. The outer surface carries faint ridges where the polyp's corallite walls fused with neighboring polyps over years of slow accumulation. The interior is porous. Tiny channels run through the carbonate in all directions, left by the tissues that once threaded through the skeleton. And within these channels, in the hairline spaces between the crystalline walls, a community of microbes has established itself. They are endolithic, stone-dwelling, and they work in the dark at a pace that has no urgency to it, processing organic matter that filters in through the pores, cycling nitrogen through chemical forms that diffuse back out into the water above. The dead branch metabolizes, in this quiet distributed way, long after the polyp that built it has dissolved. The water above that branch is cold and dark and moving steadily along the rock face. It carries particles, small organic aggregates, fragments of shell, the shed casings of copepods, mucus strands from organisms that passed through hours ago, all of it drifting at the pace the current allows, which is sometimes fast and sometimes almost still. The branch intercepts some of it. The endoliths within the branch process some of what settles into the pores, and in the space between the two ribs of carbonate, where the small creature rests, a kind of local economy is already running. This is the first thing to understand about the cold water coral reef as a place of life, rather than simply a place of coral. The framework itself, even the dead framework, is not background. It is not stage scenery against which the real biology happens elsewhere. It is habitat in the fullest sense, providing architecture, shelter, hard substrate, and a slow, continuous chemistry that the organisms sheltering within it depend on. A reef made entirely of dead skeleton continues to sustain life. It continues to be a reef. And in the darkness of these deep slopes, where bare rock would offer almost nothing, the framework offers everything. The small creature pressed between the ribs of that branch is a squat lobster, specifically Munida sarsi, a species found throughout the cold water coral reefs of the North Atlantic. Its pale orange body, the color of old bone, its front claws folded close against its thorax, its antennae trailing back along its body and held still against the current. It is perhaps the length of a large hand from the tips of its folded claws to the end of its abdomen. During the day, though day means nothing at 400 feet of depth except an abstract increment of time on a surface it has never experienced, it tends to stay inside the reef framework, tucked into whatever cavity offers the right combination of shelter and current flow. When the water accelerates, carrying denser concentrations of particles, it may move to a more exposed position and extend one claw into the flow, or it may simply wait. It does not plan this. It responds to gradients, chemical, mechanical, the faint pressure wave signatures of nearby motion. Around it, in the interstitial spaces of the same section of reef, are organisms in every direction. Isopods move in slow, deliberate traverses across the inner surfaces of coral branches. Amphipods, smaller and faster, negotiate the narrow passages between interlocked skeleton segments. Copepods hang in the still eddies behind branches where the current slows, waiting or feeding on the finest suspended particles. The diversity within a single section of coral framework, even a small section, perhaps the area of an outstretched hand, substantially exceeds that of the bare sea floor a few meters away, where the rock offers no structure, no shelter, no interstitial chemistry, nothing but exposure to the current and whatever settles onto the flat stone surface. The reef creates this diversity not through any active process of hospitality, but through the simple fact of its geometry. Three-dimensional structure in an otherwise mostly flat and featureless deep-sea environment is rare and valuable. Animals that need shelter, that need attachment surfaces beyond the flat rock, that need the specific hydraulic conditions created when flowing water meets a complex branching architecture, These animals come to the reef because nowhere else provides what the reef provides. The dead skeleton creates the same opportunities as the living colony, in some ways more so because the dead skeleton is permanent, structurally stable, colonized by the endolithic chemistry that living coral tissue keeps out, and already integrated into the sediment record of the mound. The living coral lives at the surface. The dead coral supports everything below it. Above the branching framework, perhaps a meter above the dead skeleton, where the squat lobster rests, a cluster of living Lophelia pertusa polyps extends from the reef in the current's direction. Pale white to orange tubes of calcified corallite, each holding at their open end a ring of tentacles extended into the moving water. The polyps do not illuminate the darkness. They have no bioluminescence, no photoreceptors, no organ oriented toward light of any kind. What they have are tentacles, nematosis cells packed into the tentacle surface, ready to fire on contact, and mucus. The tentacles extend in the current and sweep through the water in the slow oscillating motion of animals at work. Between the polyp clusters and draped across much of the visible reef surface in every direction are brittle stars, hundreds of them. Each one has a small central disc, perhaps the diameter of a shirt button, and from this disc extend 5 arms that are long relative to the disc, thin, flexible, and in constant slow motion. The brittle star anchors itself to the coral branch with 2 or 3 of its arms and extends the remaining arms into the current, holding them open in curves that maximize their exposure to passing particles. The arm surface is covered in small spines and tube feet that intercept what the current delivers. When a particle contacts the arm, tube feet and mucus work together to transfer it toward the central disc, where the mouth waits. In some sections of Norwegian reef, the density of brittle stars on the coral surface runs to hundreds of individuals per square meter. The entire visible surface of the coral framework, the upper surfaces of branches, the undersides of branches, the lateral faces of every available structure, is covered with these slow-moving arms. At a distance, the reef appears to be in motion, not because the coral moves, but because the brittle stars covering it never stop. Their arms drift and sway and reach and retract in the current. The whole surface of the framework, animated by this distributed, patient reaching that has no urgency and no rest. Basket stars are larger. Their central disk may be several inches across, and from it extend branching arms that subdivide again and again, each subdivision thinner than the last, until the outermost tips are fine as thread. A large basket star, arms fully extended, spreads across an area the size of a dinner plate. It anchors itself to a coral branch at the highest available point, the tip of a living colony, if it can reach it, and spreads its arms into the current as a net. When a zooplankton or larger particle contacts the mesh of the outer branches, the arms contract and curl inward, transferring the captured organism to the central disk. The basket star does not need much current. It does not need a specific chemical gradient. It needs elevation, height within the water column relative to the seafloor, and the reef provides this. Without the reef framework projecting upward from the rock, the basket star would have nowhere to anchor that gets it off the bottom and into the richer current above. The feather stars occupy the same elevated positions by different means. They grip the coral branch with grasping structures called cirri and extend their arms upward and outward, each arm fringed with long pinnules that give it the feathered appearance that names the group. The pinnules, covered in small tube feet and mucus, intercept particles passing through the water. A feather star feeding in a moderate current is a still image of patience, arms spread, pinnules extended, the animal holding its position without apparent effort, as the water delivers what it needs. None of these organisms compete with the coral for food in a way that threatens the coral. The reef framework is large enough and the current delivers enough that the suspension feeders layered across its surface can all extract from the flow without depleting it for any of the others. The reef is not a finite resource in this sense. It is an amplifier of the local food environment. The branching structure increases surface area, concentrates particles in specific hydraulic zones, and creates eddies where settling rates slow and the probability of interception increases. Every organism attached to the reef framework experiences a somewhat richer food environment than it would experience on the bare seafloor beside it, and the reef benefits from the structure these organisms add, their bodies becoming additional attachment surfaces for other recruits. Sea urchins move slowly across the base of the reef and through the sediment accumulated within it, grazing on the biofilm and organic material that accumulates on hard surfaces. They do not eat the living coral. The coral tissue, protected by its nematocysts, is not a safe food source for most urchins, but they crop the surfaces of dead framework, keeping them clean of dense biofilm that might otherwise inhibit new coral settlement. They move at the pace that urchins always move, which is very slow, and they leave behind a clean carbonate surface that is available for the next generation of settling coral larvae. Their work takes years to become visible, and nothing about it suggests intention. They eat what is in front of them, and what is in front of them is often the surface of a dead Lophelia branch. Polychaetae worms thread through the interstices of the framework, tube-dwelling species extending their feathery radials into the water above their tubes to filter feed in the same current that feeds the brittle stars and feather stars above them. Free-living species moving between coral branches in search of the small organic deposits and microbes that accumulate on dead carbonate surfaces. The worms are everywhere in this framework, inside dead coral tubes, buried in the sediment, caught between branches, tucked into cracks in the rock below the reef base. In a census of a section of cold water coral reef, Polychaeta species consistently number among the most diverse groups present, their variety of forms and feeding strategies filling nearly every available ecological position within the framework community. Sponges attach to dead coral surfaces and project upward in pale irregular masses, filtering the water column through their internal canal systems and expelling it as cleaned water from their exhalant openings. A sponge filters an enormous volume of water relative to its size, extracting bacteria, dissolved organic material, and the very finest particles that other filter feeders cannot capture. In some sections of reef, particularly where the current is slower and particles settle rather than flowing past in the water column, sponges represent a substantial fraction of the visible biomass on dead framework surfaces. Their tissues carry an unusual internal community of bacteria and archaea that contribute to the sponge's nutrition and that process nitrogen and sulfur compounds in ways that may contribute to the broader nutrient cycling of the reef system. The fish come and go, but some stay for extended periods. A tusk fish, large, pale, elongated, hovers in the water just outside the entrance to a cavity in the coral framework. Its body is perhaps the length of a grown person's arm, from tail to snout. The cavity behind it has been formed by the collapse of several dead coral branches, and the space inside is perhaps a foot deep and a little wider than that. The walls made of interlocked carbonate branches coated with brittle stars and small sponges. The tusk fish is not feeding at this moment. It is holding position with small adjustments of its pectoral fins, oriented into the current, resting in the lee of the reef structure where the water moves more slowly. The reef, from the fish's perspective, is primarily shelter, a geometric complexity in an otherwise open water column where there is little to hide behind and where predators can approach from any direction. During spawning season, tusk fish aggregate at Lophelia reefs on the Norwegian margin in concentrations that surveys conducted by remotely operated vehicles have documented consistently over years of observation. The reefs function as spawning grounds, sites that fish return to, that the fish locate through mechanisms that are not fully understood, navigating to specific sections of reef in depths where light gives no navigational information. Redfish aggregate at the same sites for the same purpose. Ling, another commercially significant species, uses the interstitial cavities of the reef framework as nursery habitat for juvenile fish during early growth stages. The concentration of fish biomass above cold water coral reef habitat, relative to the bare seafloor beside it, is measurable and consistent across multiple survey sites. Where there is reef, there are fish. Where the reef has been removed, surveys find the seafloor sparse. This relationship reflects nothing more than the basic logic of structure. Complex habitat supports more species, more feeding opportunities, more shelter, more ways of being a fish in the deep ocean. The reef does not care about the fish. The fish come because the reef creates what they need. There is a particular depth in these Norwegian reef systems where the framework transitions from living coral at the uppermost surface to a gradient of progressively older and more dead material extending downward. The living colony, a thin veneer over centuries of accumulated skeleton. At perhaps a meter below the active reef surface, the coral is decades dead. The framework has been partially colonized by encrusting organisms, bryozoans, small tube-dwelling worms, encrusting sponges, whose calcium carbonate and silica structures add to the complexity of the dead reef matrix. The coral branch itself is still intact, still structurally sound, still providing the same shelter it provided when the living polyps covered its outer surface. It is simply occupied differently now. At perhaps several meters below the active surface, it is simply occupied differently now. At perhaps several meters below the active surface, uncover, when Semelmed, the framework grades into a dense matrix of coral rubble and biogenic sediment, the ground-up remains of coral branches that fragmented over years of movement and biological processing, mixed with the shells of foraminifera, the spicules of sponges, the calcareous remains of bryozoans and mollusks and echinoderms, and the fine organic material of bacterial cells and their metabolic products. This sediment is not inert, it is inhabited. Meiofaunal organisms, animals small enough that a grain of sand is a significant obstacle, move through its pore spaces. Nematode worms, foraminifera, copods, ostracods, and a diversity of single-celled organisms occupy the sediment column within the reef framework to depths of many centimeters, sustained by the organic matter that the reef above them traps and concentrates. The sediment within a cold water coral reef framework is significantly richer in organic carbon than the sediment of the bare seafloor beside it. The reef intercepts particles that would otherwise continue moving with the current until they settled on the open seafloor much farther away. It traps them within its interstices, concentrating organic material in a small area and making it available to the infaunal community buried within. This concentration of organic material within the framework drives the active nitrogen cycling, sulfate reduction, and methanogenesis that endolithic microbes and sediment-dwelling bacteria carry out within the reef, chemical processes whose products diffuse upward through the sediment and back into the water column, where they become available for further uptake by the suspension feeders above. The reef is not a passive accumulation of calcium carbonate. It is a slow engine. Food comes in from outside, carried by the current. The polyps and the suspension feeders on the reef surface intercept some of it. The remainder, the particles that escape capture at the surface, settles into the framework and is processed by the community within it. The chemical products of that internal processing diffuse back up into the water above the reef, contributing to the nutrient field that the surface-dwelling organisms draw from. Nothing is wasted in an absolute sense, though the steps by which organic material moves through the system, from particle to tentacle to polyp tissue, from settled organic matter to bacterial metabolism, to dissolved nutrients, from dissolved nutrients to new polyp tissue, take different amounts of time and pass through different bodies at each step. Beyond the active reef, in the water column above it, nothing visible marks the boundary between reef and open water. The darkness is the same. The temperature is the same. The current that flows over the reef continues over the bare seafloor beside it without changing its character. But the water directly above a Lophelia reef carries a chemical signature that can be detected, elevated concentrations of ammonium and phosphate, the products of the metabolic activity within the framework, diffusing upward from the reef into the water above it. This chemical plume extends for some distance above and downstream of the reef, and within it, the concentration of planktonic organisms is measurably different from the concentration in the surrounding water. The reef generates, in this way, a local enrichment of the water above it that feeds back into the reef itself. The largest organisms in the reef community are also the most intermittent visitors. Sharks use the reef framework as a resting area during hours when they are not actively hunting. The specific species varies with latitude and depth. The blackmouth cat shark on European margins, other species on seamounts in the southern Atlantic and around New Zealand, but the behavior is consistent. The sharks rest on or near the reef surface, sometimes tucked into the larger cavities within the framework, sometimes lying on the flat sediment just beside the reef base, holding still in a way that observers on remotely operated vehicles have documented repeatedly. They are not feeding. They are conserving energy in the shelter of a structure that is large enough to provide a reference point in open water and deep enough to offer some hydraulic protection against the current. The reef, from the perspective of these large animals, is a landmark. In a deep ocean where the seafloor is mostly flat and featureless for miles in any direction, a mound rising 30 or 50 or 100 feet from the surrounding sediment is significant. It alters the local current pattern. It concentrates food. It provides reference points for navigation. It is, in the most basic sense, a place, a specific location in the otherwise undifferentiated geography of the deep sea floor that animals return to, that fish aggregate at during spawning, that sharks rest beside, that squat lobsters spend entire lives within. The framework that creates this place is built by an organism with no awareness of what it creates. A Lophelia polyp secretes carbonate and extends its tentacles into the current. That is all it does. Over years, and then decades, and then centuries, the accumulated secretions of many polyps, living and dying, and being built upon by successive generations, produce the three-dimensional structure that hosts this entire community. The community depends on the structure. The structure is produced by organisms that have no knowledge of the community depending on it, no orientation toward the function their secretions serve, no awareness of the squat lobster in the cavity, a few centimeters below their living surface. The cave system within a large coral mound goes deep in sections where the accumulated framework has become very dense and the internal cavities have been partially closed by subsequent growth. The spaces within are truly enclosed, dark even relative to the dark water outside them, sheltered from all current, their walls covered in organisms that prefer stillness over flow. These innermost chambers, if they can be called that, gaps between compacted coral branches that happen to have remained open over centuries of accumulating growth around them, host communities almost entirely distinct from those on the reef's outer surface. Organisms that cannot compete in the higher energy surface environment find, in these still interiors, the conditions they need. The diversity of the reef is in this way not uniformly distributed across its surface. It is stratified by flow, by exposure, by light. No, there is no light anywhere, by current velocity and direction, by the degree of enclosure, by proximity to the sediment and its associated chemistry. Every position within the reef framework has its own set of conditions, and every set of conditions has its own community, and these communities overlap and interact along the gradients between them. The result is a layering of life within a structure that at first inspection from outside might appear to be simply a pile of pale branching stone in the dark. At the outer face of the reef, where the framework is newest and most exposed to the current, the living polyps are densest, their tentacles extended in the maximum flow. The surfaces just below the active growing edge are covered in the suspension feeders, brittle stars, feather stars, basket stars, that exploit the same elevated food concentration that the coral exploits. Further back, where current velocity falls off behind the reef's bulk, the sponges are more abundant, their lower flow requirements matched by the hydraulic shadow the reef casts downstream. In the sediment at the reef's base, the infaunal community processes what has settled over years of reef activity. And within the dead framework itself, in every direction, the endolithic microbes and the mobile crustaceans and the tube-dwelling worms occupy the interstitial spaces, each in its own position within the geometry of accumulated calcium carbonate. The whole assemblage, from the surface polyp to the deepest sediment-dwelling nematode, is knitted together by the flow of materials, organic particles in, chemical compounds cycling through, waste products diffusing out, nutrients recycling upward. None of these organisms is aware of being part of something larger than itself. Each responds only to the immediate conditions of its position, the particle in the tentacle, the chemical gradient in the sediment, the pressure wave of a nearby animal, the mechanical stimulus of a food item making contact with a spine or a tube foot or a nematocyst. The larger pattern emerges from these local responses without any of the participants having access to the larger pattern. The cold water coral reef is not, in this, different from any other complex community of organisms, but it exists in a place where complexity was for a long time not expected. The deep sea was imagined, until relatively recently, as a sparse and barren environment, its darkness and pressure and cold suggesting to surface-dwelling intuition a place too extreme for much life. Surveys with baited cameras and trawls revealed that the flat abyssal seafloor does support some life, but at low densities and relatively low diversity. The discovery that cold water coral reefs host biological communities of the richness now documented, brittle star densities that blanket every surface, crustacean diversity that exceeds the surrounding open seafloor by orders of magnitude, fish aggregations significant enough to attract commercial fishing fleets, revised the picture substantially. The reefs were not known to most people who did not study them, because they exist in a part of the ocean that surface-going vessels cannot directly observe, and because the tools needed to see them, the submersibles, the remotely operated vehicles, with their lights and cameras, the acoustic mapping systems that can resolve topographic features a few meters in height on the seafloor, became capable of systematic survey only in recent decades. But the reefs themselves have been there for a very long time. Some of the mounds now being mapped with high-resolution acoustic systems began growing before the last glaciation reached its maximum. The community of brittle stars covering their surfaces, the squat lobsters in their interstices, the fish sheltering in their cavities, these are not new arrivals. They represent the continuation of an ecological relationship between reef framework and reef community that has been running in roughly its present form for tens of thousands of years. The dead framework that makes up most of any large cold water coral mound preserves evidence of all of this. Within the interstices of deep buried coral skeleton, preserved in the carbonate matrix and the sediment infilling, are the fragments and remains of communities that used the reef in past millennia, the same way present communities use the reef now. The species may differ in detail, the distribution of temperature and oxygen conditions during glacial periods shifted which organisms could live at which depths, but the functional structure of the community is recognizably similar. Suspension feeders on exposed surfaces, in fauna in the sediment within, crustaceans in the cavities, fish in the open water above. The architecture of the reef generates the architecture of its community, and this relationship has been stable across the kinds of time that make human history seem brief. A mound that has been accumulating for 100,000 years is mostly dead, calcium carbonate and biogenic sediment, with living coral only at its outer surface. The ratio of dead to living within such a mound is overwhelming. The living tissue covers perhaps a few percent of the mound's total volume, the rest of it built from the skeletons of animals that calcified their last atom of carbonate and died perhaps 10,000 years ago, perhaps 50,000 years ago, perhaps a few hundred years ago. And yet the mound supports at its surface a diverse and actively functioning biological community, sustained by the architecture that all those long-dead organisms produced. This is what makes the cold water coral mound different from most other biological structures. A forest is made of living trees, a tropical reef is mostly living coral, and the organisms associated with it, its dead skeleton representing a relatively small fraction of its total structure. A cold water coral mound is a monument built almost entirely by the dead, inhabited by the living at its surface. The living community occupies, in a sense, the very top of an enormous biological archive, using the architecture of the past to build its present. There is a quietness to this that is different from the quietness of the surrounding deep sea, which is quiet simply because there is very little there. The quietness within a cold water coral reef framework is the quietness of a place that is very full, very slowly turning, every surface occupied, every current exploited, every cavity inhabited, a biological engine running in the dark at the slowest of steady paces, with no urgency and no interruption, in water that is 4 degrees above freezing and has been dark for as long as the mound has stood. The squat lobster between the carbonate ribs does not move. The brittle stars on the outer surface of the reef above it do not stop their slow reaching. The endoliths in the pores of the dead branch process organic matter at a pace that has no correspondence to any clock a surface animal would recognize. The living polyps at the mound's peak extend their tentacles into the current and wait for what the water brings, as they always have, as the generations before them always did, in this cold and permanent and inhabited dark. The mound below them has been growing for longer than any human structure has stood. The living coral at its surface is a thin and present thing, resting on the deep accumulated evidence of everything that built it, and the community sheltering within that evidence is as old and as layered as the mound itself. Life using the work of the dead to continue in the dark without haste, without interruption, as it has for as long as the cold current has run along this slope. 4 millimeters. In the cold, dark water off the Irish margin, at a depth where the pressure of the sea above presses in from every direction, with the weight of 50 times the air at the surface, a single coral branch grows 4 millimeters in a year. Not 4 inches, not 4 centimeters. 4 millimeters, roughly the width of a pencil, roughly the thickness of 2 stacked coins, roughly the distance a fingernail grows in a slow week. That is the pace of this world. That is the speed at which stone is made from living tissue in the permanent night of the deep ocean. Hold that measurement for a moment. 4 millimeters. The branch is pale, almost white, a calcium carbonate tube secreted by a ring of soft tissue around a central mouth. The polyp inside it does not perceive the year passing. It extends its tentacles into the current when the current is strong, and it draws them inward when the current slows, and it adds its fraction of a millimeter to the skeleton beneath it in a process so gradual it would be invisible to any observer patient enough to watch. There is no urgency in this. There is no acceleration. The water is 4 degrees Celsius, and in cold like that, chemistry is careful and slow, and the carbonate lattice assembles itself 1 ion at a time from the seawater passing over it, and the branch grows, and the year turns, and the branch is 4 millimeters longer than it was. Now allow that rate to run forward across 10 years. The branch is 4 centimeters longer, a little more than 1.5 inches. It has not changed its character. It is still white, still branching, still built from the same interlocking pattern of crystalline aragonite that it has always produced. Across 10 years, the colony it belongs to has spread a little wider, recruited a few new polyps at its growing tips, sheltered a changing population of brittle stars and small crustaceans in the spaces between its branches. The seafloor around it looks the same as it did a decade ago, give or take a settled layer of marine snow and the slow rearrangement of a few sediment grains by burrowing worms. Nothing about the place announces that a decade has passed. There is no light to mark the seasons. There is no weather. There is only the current and the food it carries and the slow thickening of bone. Across 100 years, the picture begins to change. The colony has grown into something that can be called a structure, a branching framework, a meter or more across, its oldest interior sections, dying back as the living tissue migrates outward toward fresher water, the dead skeleton behind it already serving as substrate for new recruits, for encrusting sponges, for the patient colonization of surfaces by organisms that require only hard ground and cold current. 100 years of 4 millimeters per year has produced something a body could shelter behind, something large enough to cast a shadow if there were any light to cast one. From the outside, the colony looks like a small tree made of pale stone, and it is this, in every meaningful sense, a tree whose medium is the seafloor, and whose leaves are the tentacles of 1,000 polyps extended into the dark. The coral species that builds these structures goes by the scientific name Desmophyllum pertusum, though it was known for many years under its older name Lophelia pertusa, and both names refer to the same organism, a colonial scleractinian coral, a maker of stone cups, a builder of frameworks from calcium and carbon extracted from seawater. Its polyps are not particularly large. Each one sits inside a corallite, roughly the diameter of a fingertip. Each one lives out its life in water that most organisms would find inhospitable, cold, pressurized, lightless, far from any photosynthetic energy source, and each one builds, from that inhospitable water, a structure that will outlast it by centuries. That gap between the brevity of an individual polyp's life and the duration of the structure it leaves behind is one of the quiet truths at the center of this world. The living tissue matters, certainly. The polyps feed and grow and divide and reproduce and add their increment to the framework each year. But they are only the active surface of something far older than themselves. Below the living layer, extending downward through meters, and then tens of meters, and then, in the largest formations, through more than 300 meters of accumulated skeleton and trapped sediment, lies the record of every colony that came before, every polyp that lived and calcified and died and left its carbonate cup as a foundation for the next generation. The structure is not primarily alive. It is primarily a monument, and the living coral is its current caretaker. 1,000 years of 4 millimeters per year produces a vertical rise of 4 meters. 4 meters is not a mound. It is a modest thickening of the reef, a gentle upwelling of the seafloor profile. But 1,000 years is not a long time for these structures. The mounds along the European continental margin, along the Irish and Norwegian margins, along the flanks of the Rockall Bank, and the edges of the deep Atlantic, began growing not 1,000 years ago, but tens of thousands of years ago, and some of them have been building continuously for close to 2 million years. The arithmetic of 4 millimeters per year, run forward across that span of time, produces formations of a different order entirely. Some of these mounds rise more than 300 meters from the surrounding seafloor. Some span several kilometers in diameter. They are not geological features in the sense of being made by tectonic forces or volcanic activity. They are biological features made by organisms built from the carbon of seawater and the slow labor of soft tissue. But they are so old and so large that from a certain distance they resemble hills. The Belgica Mound province lies on the southwest margin of Ireland, in water between 500 and 900 meters deep. It contains more than 60 individual mounds. Some of them began accumulating their first skeletons in the early Pleistocene, which is to say, before the glaciations that covered the northern hemisphere in ice, before the sea levels that now characterize the world's coastlines had taken their present form, before the particular configuration of ocean currents that now flows along this margin had settled into its current pattern. The base of a Belgica mound is in some cases 2 million years old. The cold water coral polyp sitting at the very top of that same mound, extending its tentacles into the Irish margin's slow deep current tonight, is adding to a structure whose foundation predates the first stone tools made by human hands. This depth of time is not easy to hold in the mind at once, and there is no need to hold it all at once. It is enough to know that the mound is there, and old, and still building, and that the coral on its summit has no knowledge of the depth below it, that the polyp simply does what polyps do. In the water and the dark, adding its 4 millimeters to a tower that has been rising since before the Ice Age. The patience embedded in that image is its own kind of peace. Nothing is rushing. Nothing will be resolved by rushing. The mound has been growing across every kind of ocean climate the planet has produced in the last 2 million years, through glacials and interglacials, through shifts in current and chemistry and temperature, and it is still growing now. Whatever happens at the surface, the mound continues. The skeleton a cold water coral polyp builds is not simply an architectural material, it is a record. The carbonate lattice of aragonite that forms inside the calcifying tissue incorporates chemical signals from the surrounding seawater, signals that carry information about the temperature of the water, its chemistry, the composition of the ocean circulation that delivered it to this place. Those signals are locked into the crystal structure of the skeleton at the moment of calcification and preserved there as the skeleton hardens and the polyp continues to grow upward, leaving the older material behind as a sealed archive. Uranium enters the growing skeleton readily, absorbed from seawater during the calcification process. Thorium does not. It is excluded by the chemistry of the process, so the newly formed skeleton contains uranium, but almost no thorium at all. From the moment calcification is complete, the uranium inside the skeleton begins to decay, and one of its decay products is thorium-230. This accumulation is steady and predictable, governed by physics that does not vary. By measuring the ratio of uranium to thorium-230 in a precisely selected sample of coral skeleton, it is possible to calculate how much time has passed since that particular layer of carbonate formed. The clock runs forward from zero at the moment of calcification, and it does not stop. This technique, uranium-thorium radiometric dating, has been applied to cold water coral skeletons from many sites across the Atlantic and Pacific, and the ages it reveals extend back through hundreds of thousands of years. A coral fragment extracted from the lower levels of a mound core and dissolved into acid for analysis may return an age of 80,000 years, or 150,000, or further still. That number is not an estimate. It is a measurement derived from the ratio of two isotopes in a few milligrams of carbonate, and it places the growth of that particular skeleton at a specific moment in the planet's climate history. 80,000 years ago, the Earth was in the later stages of a glacial cycle. Ice sheets covered much of the northern hemisphere. Sea levels were lower than they are today, perhaps by 250 feet or more. The Atlantic Ocean circulated differently, its great conveyor of warm surface water and cold deep return flow running at patterns that do not match the present configuration. The deep water at the Irish margin, where the coral was growing, had a particular temperature, a particular chemistry, a particular oxygen content, all of it different from what the margin experiences today, and all of it encoded in the skeleton of a coral polyp that was simply feeding and growing in its dark cold water, building its carbonate cup with no knowledge of the planetary drama surrounding it. The polyps that build cold water coral skeletons also incorporate boron into their carbonate matrix, and the ratio of two boron isotopes within that matrix varies in a predictable way with the pH of the surrounding seawater. When ocean water is more acidic, the ratio shifts in one direction. When it is more basic, it shifts in the other. By measuring boron isotope ratios in precisely dated coral specimens, it is possible to reconstruct the pH of deep ocean water at specific moments in the past with a precision that few other natural archives can match. The same precisely dated skeletons also incorporate neodymium, a rare earth element whose isotopic signature reflects the provenance of the water mass that bathed the coral, which ocean basin it came from, which circulation pathway it traveled, how far it had journeyed from its last contact with the atmosphere. Put these records together, the age from uranium-thorium, the past ocean chemistry from boron, the circulation history from neodymium, and a single fragment of cold water coral skeleton becomes a document describing the state of the deep ocean at a specific moment in time, written in the language of isotope ratios and crystal chemistry, and legible to researchers who know how to read it. These records have been used to trace the behavior of Atlantic circulation across the last glacial maximum and the deglaciation that followed. They have shown that the great ocean conveyor reorganized abruptly during the period of rapid warming at the end of the last ice age, switching between different configurations in intervals of years to decades rather than the centuries one might expect from such a large and inertial system. They have documented moments when deep water formation in the North Atlantic slowed or stopped, altering heat distribution across the entire hemisphere. All of this history was being written, slowly, in the dark, by organisms that had no stake in the global climate beyond the temperature and chemistry of the water immediately surrounding them. The coral did not record these events intentionally. It simply calcified, as it always does, incorporating whatever was present in the water into the structure it was building. The archive is a byproduct of the growth, not a purpose of it. And that, the fact that this planetary record was created without design, as a side effect of millions of individual polyps simply doing what polyps do, carries its own quiet weight. A core extracted from a cold water coral mound contains within it layers that span different periods of ocean history, each layer growing at the pace of 4 millimeters per year, each one preserving the chemistry of the deep water at the time of its formation. At the bottom of the core, the oldest skeleton may have formed during a period when this part of the Atlantic was covered in ice at the surface and circulated in a pattern unlike anything observed in the historical record. Moving up the core, each layer represents a step forward in time: the glacial maximum, the first pulse of warming, the meltwater events that disrupted circulation, the gradual settling of the ocean into its current configuration. The coral grew through all of it, adding 4 millimeters per year, building its stone record of a changing world, never ceasing. The growth rate of 4 millimeters per year is an average, and like all averages, it conceals variation. In years when the current delivers abundant organic particles, when the temperature stays close to the optimal range, when the aragonite saturation of the water remains high enough for calcification to proceed efficiently, the coral may grow closer to 35 millimeters. In years when conditions are less favorable, when the current weakens, when the organic flux from the surface diminishes, when the water chemistry shifts, growth may slow to only a few millimeters. These variations leave traces in the skeleton, faint banding visible in thin cross-sections examined under fluorescent light, a physical record of good years and lean years laid down in alternating densities of carbonate crystal. The banding is not as pronounced as the growth rings of a tree. The seasons at depth are gentler, filtered, and time-delayed, the pulse of organic matter arriving months after the surface bloom that produced it. But it is there, in a cross-section of a Lophelia pertusa branch, held under the right wavelength of light. The alternating bands appear as pale and dark stripes in the white carbonate, each one a year in the coral's life, each one produced in total darkness by a polyp that experienced no direct sunlight at any point in its existence. The light used to reveal these bands in a laboratory is the first light to touch that carbonate since it formed on the seafloor, and the gap between those two moments may be 100 years, or 1,000 or more. There is something worth sitting with in that image. The band of carbonate, formed in the dark, illuminated for the first time in a laboratory by a researcher's fluorescent lamp, the light arriving centuries late to a record that had been quietly waiting. Not waiting in any intentional sense. The carbonate does not wait. It simply persists, as carbonate does, stable and patient, indifferent to whether it is ever read. The reading is something humans do to it, imposing a narrative on a chemical record that predates any human interest in it by a very long margin. A cold water coral mound off the Irish margin, rising 300 meters from the surrounding seafloor, has been building for 2 million years. Its base is composed of skeletons from the Pleistocene, compacted and cemented and buried under layers of subsequent growth and trapped sediment. Its lower flanks contain the skeletons of colonies that grew during glacial periods, their chemistry reflecting the cold, dense water that bathed them during those long, cold intervals. Its middle sections record the transitions between glacial and interglacial conditions, the abrupt shifts in circulation, the meltwater pulses that temporarily suppressed deep water formation, and changed the temperature and chemistry of the deep Atlantic. And its upper surface, the living surface, the few meters of active colony extending tentacles into night's current, continues to add its increment to the record, building on top of everything that came before. The mound is not aware of its own history. No part of it reflects on what it has preserved. The living polyps at the summit are engaged entirely with the present moment, the current, the food it carries, the temperature of the water, the chemical signals that trigger or suppress feeding, the slow division of cells that produces new polyps at the growing tips. Everything below them is simply the substrate they happen to occupy, the accumulated foundation of a process they are continuing without reference to its past. This is one of the deepest contrasts between this world and the world of surfaces, where memory and narrative and the awareness of history are constant companions to experience. In the cold, dark water of the deep margin, there is only the present moment of chemistry and current, and the skeleton growing outward from it, and the record being made without any recorder. The archive accumulates because the process continues. The process continues because the water is cold and carries food, and the hard substrate offers purchase. Nothing more is required. Down here, below the last reach of any sunlight, the seafloor has a texture built from accumulation. A hand moving across the surface of a cold water coral mound would feel the rough, irregular architecture of interlocked skeletal branches, some of them living, their tentacles withdrawing at the disturbance, and many of them dead. The white carbonate slightly roughened by endolithic organisms that have colonized its surface. Between the branches, the fingers would find soft sediment, fine particles that have sifted down from the water column over years and decades, becoming trapped in the framework's complex interior spaces, accumulating as a fine matrix that fills the gaps and records, in its layers the slow rain of material from above. This sediment is not inert. Within it, communities of microbes process organic material, cycling nitrogen and sulfur and carbon through transformations that contribute to the chemistry of the water above the reef. Endolithic bacteria work within the carbonate of the dead skeleton itself, their metabolic byproducts slowly altering the crystal structure of the material they inhabit, preparing it for eventual dissolution and reconstitution in new skeletal form. The dead framework is not simply architecture. It is a substrate for ongoing biological and chemical work, a medium through which matter continues to move long after the coral that built it has died. Cold water coral mounds are in this sense not dead places with living surfaces. They are living places through and through, active chemical environments, inhabited at every scale from the microbial to the meter-long, processing and cycling and transforming material in the dark with a thoroughness that has no equivalent in any other deep-sea habitat. The living coral at the summit is the most visible part of this activity, but it is not the whole of it. The mound is alive from its sediment-filled base to its outermost tentacles in ways that express themselves through chemistry and time rather than motion and color. Far above this mound, Some 800 meters of dark, cold seawater stretches toward the surface. Somewhere near the top of that column, the light begins, first as a dim blue-green suggestion in the uppermost water, then, as depth decreases, as a real presence, warming and brightening toward the surface until it reaches the sunlit zone where phytoplankton drift and photosynthesize and produce. Over the course of weeks and months, the organic particles that will eventually descend toward the reef below. The sunlit surface and the dark deep floor are connected by this slow downward movement of matter, the dead cells, the shed mucus, the fecal pellets, the aggregated organic debris that constitutes the marine snow, but the connection is indirect and time-delayed. And the deep reef experiences the surface only as a chemical signal arriving months after the event that produced it. A spring bloom in the waters above the Irish margin produces billions of phytoplankton cells over a period of weeks in April and May. Those cells die and sink, aggregating as they fall, gathering mass, slowing in their descent as finer particles coalesce into larger aggregates. By October, some portion of that organic material has reached the depth of the mound, arriving not as identifiable phytoplankton cells, but as broken down organic particles, enriched in carbon and nitrogen, indistinguishable in composition from other marine snow, except in their seasonal timing. The reef does not know this is the echo of spring. The polyps simply extend their tentacles when the organic flux increases, responding to the chemistry of the water rather than to any seasonal signal they could detect directly. The connection between the sunlit spring and the autumn feeding pulse at depth is real, but it exists only in the physics of particle settling and the chemistry of organic decomposition. It is a connection without awareness on either end. Sit with the scale of this for a moment. A coral mound, 2 million years old, built from the skeletons of generations uncountable, rising 300 meters from the seafloor in permanent darkness. Its living surface adding 4 millimeters per year to a structure that has outlasted every civilization that has ever appeared on the land above it. Alongside this mound, dozens of other mounds rise from the same margin, each of them old, each of them building, each of them preserving in its carbonate layers a continuous record of the ocean's history that extends further back in time than any human archive. The entire province of mounds, the Belgica province, the Rock-Ultrauf mounds, the Norwegian structures, the deep Atlantic and Pacific formations, forms a distributed library of ocean history, written in chemistry, stored in darkness, largely unread. The reading has begun. Researchers extract cores from these mounds and dissolve measured fragments of their skeletons in clean acid and run the resulting solutions through mass spectrometers that measure the ratios of isotopes with a precision that would have seemed impossible a few decades ago. From those measurements, they extract ages, temperatures, circulation histories, pH records, the chemical biography of the deep ocean at specific moments across the last 2 million years. The mounds yield this information without diminishment. A fragment dissolved for analysis is gone, replaced by data, but the mound itself continues to grow, continues to add new skeleton to its summit, continues to extend the record forward into the present and beyond. The forward extension is what the living coral provides. Every year, the polyps at the summit of a Belgica mound add their 4 millimeters, or 10, or 20, depending on conditions, to a record that will eventually be readable by someone who has not yet been born, using techniques that have not yet been developed, asking questions about the ocean that have not yet been formed. The coral does not provide for this future reading. It simply grows as it has always grown in the dark and the cold, adding carbonate to carbonate, carrying forward a process that began before the glaciations, before the first human fire, before anything that could be called human history in any sense. The patience of this process is total. It does not require witness. It does not require understanding. It has been proceeding in exactly this way, the polyp extending its tentacles, capturing its food, calcifying its skeleton, dying and leaving its cup behind for the next generation, across an interval of time that contains all of recorded human history, many hundreds of times over. Long before anyone knew these mounds existed, they were growing. Long before anyone had the tools to read the records they preserve, those records were being written, and they will continue to be written long after the present moment has passed into whatever the deep future holds, because the cold water of the ocean margins will still be there, and the current will still flow, and there will still be food in it, and the coral will still grow. The upper surface of the mound is where all of this is most immediate. The living polyps there, pale, soft-bodied, each one housed in its small carbonate cup, are the present moment of the mound's long life. They are connected to every polyp that came before them through the unbroken continuity of the skeletal framework, each generation building on the foundation laid down by the last, the structure accumulating beneath them as they grow. They have no knowledge of this depth below them. They live entirely in the present tense, the current, the food, the chemistry of the surrounding water, the slow elaboration of the calcifying tissue that produces 4 millimeters per year, the future substrate for whatever comes after them. What comes after them is more coral, more skeleton, more sediment filling the expanding interstices of the framework, more time passing in the dark. The mound will grow, provided the water chemistry remains within the range that makes calcification possible. And even if conditions change, even if a portion of the living surface dies back, the dead framework persists, waiting for the conditions that will allow new recruits to settle and begin again. Cold water coral mounds have survived glaciations, sea level changes, shifts in ocean circulation, and intervals of reduced organic flux from the surface. They are among the most durable biological structures on Earth, not because they are hard in the sense of being impervious, but because they are patient in the sense of being willing to wait across any interval for conditions to return. Patience is perhaps the wrong word. Patience implies a subject waiting, aware of the passage of time, able to experience the interval as long. None of that is present here.